This is Lab Report 9, Biochemical Test 3. This is Group 2, Diego Castillo and Rita Cantu Mahurin, myself. In our introduction, the purpose highlighted in this lab was to identify and differentiate bacterial species based on key enzymatic activities and antibiotic susceptibility patterns. We utilized the catalase oxidase and nitrate reduction tests, as well as the Kirby-Bauer susceptibility assay. And we observed the metabolic diversity as well as resistance mechanisms that are critical for bacterial classification as well as clinical treatment decisions. The objectives summarized were to determine the enzymatic presence, evaluate bacterial sensitivity, and also compare the biochemical profiles of known and unknown organisms. The materials we used as far as media and cultures included the nitrate broth and the blood agar plates, and the organisms were the A. facialis, E. coli, P. aeruginosa, SRS, S. epidermis, S. saffroticis, and then the S. pyogens, as well as the S. agilite. And then our unknown number 1 and number 2, unknown number 1 is 112, and then unknown number 2 is 292. The reagents and supplies we used were the glass slides, petri dishes, the 3% hydrogen peroxide, the oxidase reagent, sterile cotton swabs, and then we also utilized the sulfonic acid, alpha naphthylamine, zinc dust, and then the bacitrine, novobiosin, and optochin antibiotic discs. For the procedure, in the catalase test, we transferred the bacterial colony to a glass slide. We added a drop of the 3% hydrogen peroxide, observed for bubbling, and then we avoided blood contamination when we were sampling it from the blood agar. The oxidase test, we swabbed a colony with a sterile cotton swab. We added 2 to 3 drops of oxidase reagent directly onto the swab, and we recorded the color change within 20 seconds. The nitrate reduction test, we inoculated the nitrate broth and incubated it, and then we added sulfonamic acid and alpha naphthylamine. And if no red color developed, we added the zinc and we observed the reaction. The antibiotic susceptibility test, we created the inoculations on blood agar plates, and then we placed the antibiotic discs accordingly as listed below, and then we incubated and measured the zones of inhibition when we were measuring the results.
These are the results for the catalase test. First, we have SRS, which there is apparent bubbling. S. pyogens has no clear bubbling. Both the unknowns appeared to show slight bubbling, but it wasn't as active as the SRS. Um, if there's immediate bubbling, like there is an SRS, that means there's catalase breaking down hydrogen peroxide into water and oxygen. So it means it's an aerobic or a facultative anaerobe. And then for the S. pyogens, that's negative. There's no bubbling, meaning it lacks that catalase enzyme, and it means it's an aerotolerant anaerobe. For the unknowns, we can maybe say they did bubble, so that could mean they are catalase present. And if they have bubbling, that helps differentiate staph from strep, and if they're positive, that's usually a staph. Um, the immediate bubbling is that hydrogen peroxide. To the hydrogen peroxide is just the changing of that water and oxygen, as I said. And when hydrogen peroxide is poured onto an open wound, that's why there's bubbling, because catalase in the cells will break down that hydrogen peroxide into that water and oxygen. And the bubbling is that oxygen gas, and it's known as a enzymatic decomposition reaction. These are the oxidase test. The E. coli came out negative. P. aeruginosa came out as a positive because it's purple. The unknown number 1 or 112 also came out positive just from the slight purple color. And the unknown 292 came out negative. The E. coli came out negative because there's no color change, meaning that it lacks the cytochrome C oxidase. And so that means it uses different ETC enzymes. The P. aeruginosa is a positive because there is a purple color that appeared within those 30 seconds, and it has a cytochrome C oxidase. And that means that it's oxidase positive, meaning there's aerobic respiration. And the same thing goes for unknown 112. Since it turned purple, it means it's a positive, meaning it has cytochrome C oxidase, and it'll be an aerobic respiration. As for unknown number 2, it did not change, so that means it is a negative. These are the results for the nitrate test. Our E. coli changed to that red color with just the reagents. No zinc was added, meaning that it is a nitrate reduced to a nitrite, and E. coli has nitrate reductase. For the P. aeruginosa, it did not change with just the reagents. It changed to a pink color after zinc was added, and that means there was a reduced nitrate beyond nitrite, and there was a complete nitrate reduction. The A. fascialis was a negative, and it turned a pale pink after adding the zinc, um, meaning that it uh, was reduced, the nitrate was reduced to nitrite by the zinc, not by the bacteria itself. So that means there is no nitrate reduction by the organism itself. For the unknown, they both became pink after the reagents were used and no zinc was used. So that means they are both positive, meaning that there is a nitrate reductase in both of them. The purpose of adding that zinc is to confirm a true negative test, because if that nitrate is still present, zinc will be reduced to nitrite, that red color, through that zinc, and it's a non-enzymatic chemical reaction. These are the results for the antibiotic test. Both bacitracin and optogin were used in the S pyogens, and it only was sensitive to the bacitracin, and meaning that the cell wall synthesis was inhibited by the bacitracin, and it helps to ID it from S. agalactae. For the S. agalactae, it was resistant to the bacitracin. However, our test was a little unconventional, and for some reason there was no growth anywhere except two spots, which can be seen on the left side. So our results were a bit skewed. Maybe there wasn't enough streaking, but it should have been resistant to the bacitracin, and it lacks that sensitivity to it, and it helps to differentiate from the aspiogens. For the S. epidermidis, novobiocin was used, and it is sensitive to that novobiocin, which is why you see that circle around it, where the bacteria did not grow, because their DNA gyrase is inhibited, and it helps to ID it from S. saprophyticus. As for that one, S. saprophyticus was novobiocin, and it's resistant to novobiocin, so the DNA gyrase shouldn't have been affected. There still appears to be a circle around the antibiotic, meaning that it did work effectively. However, as you can see from the difference in circle size, the S. epidermidis was around, I think, 6 centimeters, and the S. saprophyticus was about 3 or 4 centimeters. Um, The susceptibility patterns helped in identification and differentiation because S. pyogenes is bacitracin sensitive, unlike S. galacticae, which is resistant. And then the S. epidermidis is novobiocin sensitive, and S. saprophyticus is resistant. And these patterns will help us distinguish these species that are closely related. And the clinical implications of these antibiotic susceptibility testing is to help guide effective treatment by identifying which antibiotics will actually work, and it helps to detect resistance, to avoid ineffective drugs, and the resistance to bacitracin, novobiocin, or optogen can complicate treatment and delay diagnosis and necessitate alternative therapies.
In conclusion, these biochemical tests successfully distinguish bacterial species through observable enzymatic activity as well as antibiotic resistance profiles. So the catalase test identified the catalase positive the Staphylococcus versus the catalase negative Streptococcus. And then we also saw with the oxidase test that the oxidase positive P. aeruginosa was different from the E. coli. The nitrate test, we confirmed that the nitrate reduction capabilities were different among the gram-negative species. And then the antibiotic susceptibility, we noticed that there were key diagnostic differences. such as the S. epidermis sensitivity to the novobiosin versus S. saprophytosis resistance. Another important element to highlight was the clinical significance. These assays provide essential diagnostic tools for identifying bacterial pathogens as well as determining effective antibiotic treatments in the clinical setting. And so a vital step in combating this antibiotic resistance is ensuring that there is accurate patient care. Thank you so much for listening. Once again, this is VTPB 426 Group 2, Diego Castillo, and myself, Rita Cantu Mahurin.